Finally, enzyme induction can sometimes be used for our benefit, not just as a problem. A classic example is phenobarbital in neonatal jaundice. In newborns, the liver enzymes that conjugate bilirubin are immature and work slowly. So bilirubin accumulates and jaundice appears. When we give phenobarbital, it induces these liver enzymes, making them work faster. As a result, bilirubin is conjugated more efficiently, cleared from the body more quickly, and the jaundice improves. Many drug toxicities do not occur because of overdose, but because of Cytochrome P450. That's why, in today's lecture, we'll be discussing one of the most important topics in pharmacokinetics, biotransformation, with a special focus on the Cytochrome P450 enzyme system. So, let's get started. When we administer drug in the body, and as you already know, there are a variety of ways to give drugs, with the most common route being the oral route. Once given, drugs are absorbed into the plasma, which allows them to enter the bloodstream and be distributed to various tissues. A drug may bind to plasma proteins or become stored in certain tissue sites, and ultimately, we hope the drug interacts with its receptor, which determines its mechanism of action. Many drugs are then metabolized. The liver is the primary organ responsible for drug metabolism. Although it is not the only one, drugs are finally excreted, with the kidneys being the main organ. But again, not the only route. All of these processes together make up the pharmacokinetics of a drug. And in today's discussion, we will primarily focus on drug metabolism. And in metabolism mainly, we discuss the Cytochrome P450. The body treats most drugs as foreign substances, called xenobiotics, and tries to inactivate and eliminate them through various biochemical reactions. This process converts non-polar lipid-soluble drugs into more polar water-soluble compounds so that they can be easily excreted through the kidneys and are not reabsorbed. The most important organ of biotransformation is the liver. Drugs are also metabolized, though to a smaller extent by the kidneys, gut mucosa, lungs, blood, and skin. And what are the consequences of biotransformation? Largely, biotransformation inactivates drugs, and most drugs are converted into inactive metabolites. Biotransformation not only inactivates the drugs, but also convert a drug into metabolites that are also active or even more active than the parent drug. For example, diazepam to oxazepam. Such generation of active metabolites prolongs the duration of action of the drug. Biotransformation may also convert an inactive drug, that is, prodrug, into an active drug. For example, levodopa to dopamine. Chemical reactions of biotransformation occur in two phases, Phase 1 and Phase 2. Phase 1 reactions involve the reactions mediated by Cytochrome P450 systems, which include reduction, oxidation, and hydrolysis, which results in formation of polar, water-soluble metabolites, which then easily be excreted by kidneys. Phase 1 reactions is first lost in geriatric patients. Now, let us move to Phase 2 reactions, which are also called conjugation reactions. In these reactions, the body attaches endogenous water-soluble substances such as glucuronic acid, sulfuric acid, glycine, or glutathione to the drug or to its Phase 1 metabolite. As a result, a highly polar conjugate is formed. These conjugates are usually pharmacologically inactive and are readily excreted by the kidneys. Phase 2 reactions include several conjugation processes such as methylation, glucuronidation, sulfation, acetylation, and glutathione conjugation. For example, methylation involves the addition of a methyl group to the drug molecule. Glucuronidation, which is the most important Phase 2 reaction, involves the addition of glucuronic acid to the drug. Not only drugs, but also endogenous substances such as bilirubin and steroid hormones undergo glucuronide conjugation. In this process, the drug or its phase 1 metabolite combines with uridine diphosphate glucuronic acid. The transfer of glucuronic acid to the drug is catalyzed by the enzyme UDP glucuronyl transferase. The resulting drug glucuronide conjugate is highly polar, pharmacologically inactive, and can be easily excreted through the kidneys. In newborns, the enzyme UDP glucuronyl transferase is not adequately developed. As a result, bilirubin cannot be conjugated efficiently, leading to its accumulation and causing neonatal physiological jaundice. Gray baby syndrome is a serious adverse effect seen with high doses of chloramphenicol, a broad-spectrum antibiotic in neonates. This occurs because newborns lack sufficient UDP glucuronyl transferase, making them unable to metabolize and eliminate chloramphenicol effectively. The drug accumulates, leading to toxicity characterized by cyanosis, hypotension, and a grayish discoloration of the skin. Acetylation, which involving the addition of an acetyl group to the drug molecule. An important feature of acetylation is that it shows genetic or genotypic variation in the population. Based on this variation, individuals are classified as fast acetylators and slow acetylators. In fast acetylators, the drug is metabolized rapidly, leading to faster clearance from the body. In contrast, slow acetylators metabolize drugs more slowly, which can result in drug accumulation and an increased risk of adverse effects. Clinically, this is very important because slow acetylators are more prone to drug-induced SLE. Classic examples include hydralazine, a vasodilator used in severe hypertension, procainamide, an antiarrhythmic drug, and isoniazid, an important antitubercular drug. Because these drugs are metabolized slowly by slow acetylators, their accumulation in the body can trigger autoimmune-like reactions, leading to drug-induced SLE. Now let us move on to the Cytochrome P450 system, which plays a central role in phase 1 reactions of biotransformation. These enzymes are located in the microsomes, which line the smooth endoplasmic reticulum of liver cells. Cytochrome P450 represents a large family of enzymes responsible for the oxidative metabolism of a wide variety of drugs. The synthesis and activity of these microsomal enzymes, especially CYP450, can be modified by drugs and environmental pollutants. When certain substances increase the production or activity of these enzymes, the process is called enzyme induction. On the other hand, when some drugs reduce the activity of CYP450 enzymes, the process is known as enzyme inhibition. Common examples of enzyme inducers include carbamazepine, alcohol, in fact chronic alcohol use, rifampicin, griseofulvin, phenytoin, smoking. A useful mnemonic to remember enzyme inducers is CARGPS. Examples of enzyme inhibitors include cimetidine, chloramphenicol, macrolide antibiotics, especially erythromycin. Note, azithromycin has minimal to no CYP450 inhibitory effect. Ketoconazole, ciprofloxacin, antiviral drugs, particularly HIV protease inhibitors that end with avirs. Grapefruit juice. You only need to clearly remember either enzyme inducers or enzyme inhibitors, because knowing one automatically helps you infer the other in clinical situations. So, drug metabolizing enzymes, mainly the cytochrome P450 enzymes, determine how fast a drug is broken down in the body. When a drug induces these enzymes, it increases enzyme activity in the liver, causing the drug to be metabolized more rapidly. As a result, the drug disappears from the body faster. In contrast, when a drug inhibits these enzymes, it blocks or slows down enzyme activity. This causes the drug to remain in the body for a longer time, leading to higher drug levels, which may result in toxicity or increased side effects. When a patient takes a drug along with an enzyme-inducing drug, the first and most important event is increased synthesis of liver microsomal enzymes. Because of this, the liver starts working faster than normal. As a result, other drugs are metabolized more quickly, which leads to a shorter duration of action and sometimes therapeutic failure. A classic example is oral contraceptive pills failing when given with rifampicin, as rifampicin, which is enzyme inducer, result in increase the metabolism of contraceptive pills. At the same time, faster metabolism can sometimes produce more toxic metabolites, which explains why paracetamol toxicity becomes more likely in patients taking enzyme inducers. Paracetamol is metabolized into a highly toxic compound called N-acetyl P-benzoquinone imine. Under normal conditions, this toxic metabolite is immediately neutralized by glutathione and causes no harm. But in situations such as overdose, chronic alcohol intake, or enzyme induction, excessive NAPQI is produced and glutathione gets depleted. Once glutathione is exhausted, NAPQI starts binding to liver cell proteins, leading to hepatocellular injury and liver necrosis. Enzyme induction also increases the breakdown of vitamin D, leading to vitamin D deficiency, and over long periods, this can result in bone diseases such as rickets in children and osteomalacia in adults. Another important point is autoinduction, where a drug increases the metabolism of itself. For example, carbamazepine induces its own metabolism, so over time, a higher dose is required to maintain the same therapeutic effect. The response to drugs can also become unpredictable in certain populations like smokers and chronic alcoholics, because their liver enzymes are already induced. Finally, enzyme induction can sometimes be used for our benefit, not just as a problem. A classic example is phenobarbital in neonatal jaundice. In newborns, the liver enzymes that conjugate bilirubin are immature and work slowly, so bilirubin accumulates and jaundice appears. When we give phenobarbital, it induces these liver enzymes, making them work faster. As a result, bilirubin is conjugated more efficiently, cleared from the body more quickly, and the jaundice improves. So overall, enzyme induction mostly causes reduced drug effect, dose adjustments, nutrient deficiencies, unpredictable responses, and in a few situations, it is deliberately used for clinical benefit. When a patient receives a drug along with an enzyme inhibiting drug, the key event is reduced activity of liver microsomal enzymes, mainly the CYP450 system. Because of this, the liver's ability to metabolize drugs becomes slower than normal. As a result, drugs that are normally cleared by these enzymes start accumulating in the body, leading to higher plasma levels and prolonged duration of action. Clinically, this is dangerous because it increases the risk of dose-related toxicity, such as warfarin causing excessive anticoagulation and bleeding when combined with enzyme inhibitors like macrolides or azole antifungals. Similarly, phenytoin levels may rise, leading to signs of toxicity such as nystagmus, ataxia, and confusion. Enzyme inhibition can also cause unexpected adverse effects, especially with drugs that have a narrow therapeutic index, where even a small rise in drug level can be harmful. Unlike enzyme induction, inhibition usually occurs rapidly, often within a few days, because it involves blocking existing enzymes rather than making new ones. Another important consequence is that drug effects become stronger and longer lasting, so dose reduction may be required to avoid toxicity. This is a very important table. It is important not because you should sit down and memorize every entry, but because it helps you understand how the Cytochrome P450 system behaves clinically. Let's start with what actually matters. These are some of the most clinically important Cytochrome P450 enzymes and the ones that appear most often in exams and real practice. These are the few substrates that are metabolized by different cytochrome enzymes. You do not need to memorize long lists of substrates. What is important and what you must understand very clearly is the concept of enzyme induction and enzyme inhibition. Let's start with enzyme induction. A classic example, and one you should absolutely know, is cigarette smoking. Cigarette smoke contains hydrocarbons. In a smoker, these compounds stimulate the liver to produce more CYP enzymes, especially CYP1A2. So what does that mean clinically? It means that any drug metabolized by CYP1A2 will be broken down faster in a smoker. As a result, plasma drug levels will be lower. Suppose you have a patient who smokes and is taking theophylline, because smoking causes chronic airway disease and theophylline is a bronchodilator, a drug metabolized by CYP1A2. Compared to a non-smoker, this patient will metabolize theophylline more rapidly because smoking induces cytochrome enzymes which metabolize theophylline, leading to lower plasma concentrations. Therefore, smokers often require higher doses of theophylline to achieve a therapeutic effect. So now imagine this. Your patient is a smoker on theophylline, and you've already adjusted the dose upward. Later, you successfully convince the patient to stop smoking. What happens next? Well, CYP1A2 activity decreases, theophylline metabolism slows, and plasma levels rise. If you fail to reduce the dose, the patient is now at risk for theophylline toxicity. So you must be able to think in both directions when dealing with enzyme induction. On the other hand, inhibitors reduce enzyme activity, which means less metabolism of the substrate and higher plasma drug levels. Consider a patient with chronic bronchitis. These patients are often on multiple medications. The main aim of the treatment is bronchodilation. And it wouldn't be a surprise to find this particular patient on the drug theophylline. Patients with chronic bronchitis also frequently develop recurrent respiratory tract infections. And when that happens, they often require antibiotics. A common choice for respiratory infections is a macrolide antibiotic. Now here is the key question. Macrolides are potent inhibitors of cytochrome P450 enzymes. What happens when a patient on theophylline is started on a macrolide? Theophylline metabolism decreases, plasma levels rise, and the patient develops theophylline toxicity. This is a classic high-yield scenario. So remember, don't memorize the table. Understand the patterns. Know what inducers do, know what inhibitors do, and be able to predict how plasma drug levels will change. That skill will serve you far better than rote memorization.